In this short video, I'm going to be talking to you about the Golgi body. The Golgi body is involved in modification and packaging of proteins. Once a protein has been synthesized on the rough endoplasmic reticulum, it's packaged up into a vesicle, and that vesicle is transported through the cytoplasm where it fuses with the cis face of the Golgi apparatus. Within the Golgi body, it can then be modified and then released to perform a function. So we're going to talk in this video first about the structure of the Golgi body and then about its functions in a little bit more detail. The Golgi body is made up of a series of flattened sacs. It has two faces. The cis face of the Golgi apparatus faces the nucleus, and incoming transport vesicles are fused onto the Golgi apparatus at the cis face. The trans face of the Golgi apparatus faces the cell membrane, and outgoing transport vesicles bud off from the Golgi body at the trans face. The space in between the membranes are known as the lumen. Each of the flattened sacs are known as cisternae. So let's talk through each of the potential functions of the Golgi body in turn. Number one, they can modify proteins by adding carbohydrates to form glycoproteins, for instance antigens, or some hormone like HCG, human chorionic gonadotrophin, which is the hormone that is secreted by the growing placenta when a woman gets pregnant. Or clotting factors such as fibrin and thrombin that are important for making scabs if we cut ourselves. So they can be involved in modifying proteins and adding carbohydrates to form glycoproteins. The other thing that they can do is they can produce and package secretory enzymes. So let's just think about that. The enzyme would be synthesized on the rough endoplasmic reticulum, and then it would be transported via an incoming transport vesicle where it would fuse with the cis face of the Golgi body, and then within the Golgi body all these different digestive enzymes might be packaged up and then put into a vesicle here in an outgoing transport vesicle, and then that vesicle could be exocytosed out of the cell. For instance, we produce digestive enzymes in the wall of our stomach, in the wall of our small intestines, and also in our pancreas. So I just used this word exocytosis. I'm not sure how familiar you are with this term, but exo meaning exiting and cyto the cell. So exocytosis is when molecules are secreted out of the cell. Let's just imagine that here. Let's just imagine that here is our cell membrane. The vesicle would move towards the cell membrane, and then when it reaches the cell membrane, it would fuse with and release its contents out. And then the membrane that made up the wall of the vesicle now becomes part of the cell membrane. That's something that we learned about when we learned about the cell membrane, that the cell membrane is flexible and self-sealing, so things can join it and things can separate from it. Number three, it can package and secrete carbohydrates, such as those used in making plant cell walls, like cellulose and like pectin. Number four, it can transport, modify, and store lipids. Now, this is sort of a secondary function of the Golgi body that's not really to do with modification, packaging, and transport of proteins. And you'll learn about this in much more detail when you do lipid digestion in the digestion and absorption topic. It can form lysosomes. Now, lysosomes are single membraned vesicles within the cell that contain digestive enzymes like proteases, carbohydrates, lipases, and they sometimes contain lysozyme, which is an enzyme that can digest bacterial cell walls. The purpose of lysosomes is manyfold. Most commonly, it is found in all our cells, and when cells are about to undergo apoptosis, which is programmed cell death, You don't need to know that word right now, but that's what it means, apoptosis, programmed cell death. The lysosomes will burst, releasing their digestive enzymes and digesting the content of the cell. In a slightly more sinister way, they just do general housekeeping and upkeep within the cell. So say you've got an old mitochondria or an old chloroplast that you don't want anymore, the lysosomes can fuse with that organelle and then digest it. They can also form vesicles containing neurotransmitters, for instance in nerve cells. So you probably learnt at GCSE that if we imagine a nerve cell with its axon and then its axon terminal, the electrical impulse travels down the axon, and then when it reaches the axon terminal, it triggers the release of a chemical message. And that chemical message is neurotransmitters. So when the electrical impulse arrives at the axon terminal, it causes these vesicles to move towards and fuse with the cell membrane. And when they do that, they exocytose their contents into the cleft. And then that chemical message can be transferred to the next neuron, where it can trigger another nervous message to travel along the next neuron. So in this video, we've learned quite a few new key terms. So we've learned the names of the different parts of the Golgi apparatus, like the cisternae, the lumen, the cis and trans face. We've also learned a few different functions of the Golgi body, and I've introduced you to this term exocytosis and apoptosis. It's really important to keep track of new terms that you learn as you're going through the A-level biology, because biology really is a whole new language and it can get really confusing, but as long as you can understand the words, you can understand the concepts and the processes. And that's basically everything you need to know about the Golgi body for now. Well done.